Hi, my name is Kevin, and I'm a Stanford PhD student in the Department of Computer Science. I am co-advised by Dr. James Zhou and Dr. Howard Cheng. Today, I'll be talking about BABEL, a novel neural network that we have developed to enable cross-modality translation of multi-omic profiles at single-cell resolution. Single-cell multi-omics is an incredibly powerful tool, as it allows us to understand precisely how multiple layers of biological information interact. However, these methods often come at greater cost, can be more challenging to analyze due to noise, and may not always be viable depending on how samples have been stored. To address this, we developed BABEL, a deep learning model that infers a multi-omic view of individual cells, given only a single measured modality. BABEL is trained on single cells with jointly profiled expression and chromatin accessibility, and can bidirectionally infer one from the other at single-cell resolution, thus computationally enabling multi-omic profiles from only a single measured modality. To demonstrate this, we trained BABEL on a set of human cells and applied it to a set of peripheral blood mononuclear cells, or PBMCs, profiled using just single-cell ATAC-seq. The UMAP plot on the left shows the various cell types in this tissue sample. We then use BABEL to infer single-cell expression, and again use UMAP to visualize BABEL's imputations, shown here on the right. Notice how BABEL's imputations retain global relationships between cell types, despite not being given any such cell type information during its training. Also note that although this dataset exhibits substantially different noise profiles from BABEL's original training data, we do not need to correct for this when generating these predictions. Beyond overall intercellular relationships, we can also show that BABEL predicts correct overexpression of specific marker genes for specific cell clusters. The plot on the left shows BABEL's imputed expression from the previous slide, while the plot on the right is a reference plot of empirically measured single-cell expression, also in PBMC cells. If we highlight the expression of CD14 in both of these plots, we observe that BABEL's imputations on the left mirror empirical enrichment in CD14 monocytes on the right. The same goes for markers for other cell types as well, including GNLY, a marker for natural killer cells. We can also perform this attack to RNA translation in the more challenging scenario, basal cell carcinoma, or BCC. The plot on the left shows a set of BCC cells originally profiled using single-cell ATAC-seq by Yost et al. In this original work, the authors used gene activity scores to estimate expression profiles for these cells. This is a common methodology for single-cell ATAC-seq analysis. Compared to these original estimates, BABEL generates more fine-grained expression predictions given the same accessibility profiles as a starting point, despite having never seen such BCC data during its training. For example, for interferon gamma, which we expect to be localized to CD8 activated T cells, gene activity scores shown on the right are somewhat non-specific, whereas BABEL produces more localized predictions. We observe similar improvements for KLRC1, a gene that we expect to be specific to natural killer cells. Here, gene activity scores also overpredict expression in epigenetically similar cytotoxic CD8 T cells, a small error that BABEL does not make. BABEL is also easily extensible. To show this, we incorporated paired PBMC single-cell gene expression and protein epitope data to BABEL and evaluated BABEL's ability to predict protein epitopes not from expression, but from single-cell PBMC accessibility profiles, illustrated here on the left. Looking at BABEL's predicted CD16 protein abundance, we see that it correctly captures expected enrichment in CD16 monocytes and in natural killer cells. For the CD14 protein, we also see that BABEL correctly recapitulates overexpression in CD14 monocytes. As BABEL was actually never given such accessibility of protein examples, this really demonstrates BABEL's flexibility in linking across multiple different layers of molecular information. Thank you very much for your attention. If you would like to learn more, please check out our manuscript or our code base and enjoy the rest of the conference.